Hello and welcome to today's webinar titled Mapping of PAT Tools for Enabling Integrated DSP in Continuous Biomanufacturing, hosted by Biopharma Asia magazine and presented by Dejan Arzenzeg, Principal Scientist at Novartis. Hi Dejan, thanks for joining us today.
Hi.
And this presentation will be followed by Marianne Quayler, Life Science Product Manager at Kaiser Optical Systems Inc. Hi Marianne, thanks for joining us today as well.
Hello.
My name is Marcus and I will be the moderator. So before we begin, I would just like to inform our viewers that there will be a live question and answer session after both presentations near the end of the webinar. So audience members, you can send your questions at any time during the webinar via the questions tab, and that's located directly below your webinar screen. Please also state in your questions which presenters they are for. Now, please allow me to introduce our first presenter. Dejan has spent the last 10 years working for the biopharmaceutical company LEKDD, a Novartis company. Currently, he is working as a principal scientist in downstream process development department. With a PhD degree in physics, Dejan has a strong background in physical principles of protein association aggregation, along with additional skills in mathematical modeling, statistical analysis, and experimental biophysics. Dejan is currently responsible for new technology development and implementation in downstream processing. Majority of his work responsibilities is in the next Biopharm DSP project of EU Framework Programme for Research and Innovation Horizon 2020, for which the goal is to drive drug process development towards continuous processing in biopharmaceutical manufacturing. Dejan is leading new technology project of Process Analytical Technology, PAT, as part of Quality by Design approach implementation on integrated processes using advanced process control strategies where APC control system is building. I'll now be handing over to our first presenter, so welcome, Dejan.
Yeah, welcome and thank you for participating on discussing and listening to my talk. So today I will try to briefly explain recent works of the previous years on the European founded project Next Biopharm DSP, already presented at previous moment. But the work was, the project finished previous year, but the work still continues. Yeah. So for us, we are still exploring the options, how to assess the techniques needed for enabling the final goal. So to have the integrated DSP and of course the continuous biomanufacturing. So briefly, overview the content for today. So I will just start what we are doing, what we need to go towards continuous processing, our vision of process technology, techniques on DSP. Especially, it's really important which quality attributes we are trying to monitor and control, and of course then I will go further to see which techniques we assessed, and mostly today I will discuss techniques and tools going in the direction of the inline tools. Then I will show some of the cases of assessment, especially on the downstream for the chromatography steps and the final UFDF, so buffer exchange and concentration step. So just briefly, we as part of Sandoz and Novartis company, we mostly develop the biosimilars, and as a Novartis division, the main focus for us is, of course, biosimilarity, and the product quality it's really the first objective which we are really need to focus on. And of course we are exploring new techniques, so how we can tightly control and assure this biosimilarity, which is really important for the biosimilars. So of course on this path we have many challenges. So of course, for all the biomanufacturing projects and products and productions, the main focus, to reduce the costs and increase productivity. For us, especially for biosimilar companies, is to ensure biosimilarity to originators, and later on, we also have the limitation of the current process steps development. So we have the batch process development, and for these batch process steps, we have many limitations. Like in case of UFDF, it's really high. To efficiently obtain high final protein concentrations and viscosity. And at the same time, when we would like to address and assure tight controls of the product quality, we need to address the process variability. And of course, it's really hard with existing technology and analytical techniques to assess this in the right time, so at the real time. So then is the question of the advanced process control needed for that. So already assess the problem of the real-time product quality attributes monitoring, and yeah, our vision is at the end also to have and going in the direction of the real-time product release. So when we were exploring in the past and still exploring the options, of course, there are new trends coming. So one of the trends is, of course, the continuous or integrated processing on the downstream integrated processing, and the techniques for real-time product release. So with these techniques, yeah, we can be more flexible, faster, so at the same time cheaper. So we are integrated, so we can address all of the challenges regarding that mentioned above. And of course, we can go to higher productivities. But there are these challenges which I would like to address today. So the lack of inline sensors, especially for more complex quality attributes like aggregates or even more, so like host cell proteins, and so on, not only the concentration of our product, and of course we also have then the kind of contamination problems if we have the tools and the systems are not closed. So briefly, what I was meaning, with these new strategies for the biomanufacturing. So currently we have the discrete steps, but to go toward the fully integrated continuous process, there is a step-by-step approach. So because all the technologies of the process technologies, they are not ready yet. So during our assessment, there are different approaches, so-called hybrid processes, and the things are already available on the market today. But how to ensure this fully integrated continuous processes is to use, let's say, not quite new concept. So basically this concept of process analytical technology is quite old now. So even from the beginning of 2000, initiated by the FDA, and the main goal was to ensure quality by design approach, and to assess not only an understanding of what's happening of our process, but also to use it at the end for final control, for adaptive manufacturing. And this is really important when we are talking about the integrated process steps. So just really shortly and briefly, what we are talking about. So we have this QBD, so quality by design approach, which is supported by PAT. So with this approach, we assess the design space, and if we can measure the quality attributes in the real time, the PAT tools can help us for that, and with that we can design the operating space and a basic understanding between the process parameters and the outputs, which are basically the quality attributes and performance indicators. So what's then our vision? Yeah, so today we are here, so that's quite normal. So we have the discrete process steps with whole tanks between, and really to ensure the integrated approach, so and connected approach, we need inline measurements between each steps. So these inline measurements can feed in our control strategy, so for feedback and feed-forward control. So basically, without these tools, this won't be really possible. So and for that, we need undisturbed process flow. And so, of course, these inline tools needs to be in such a way that it doesn't influence your process flow. And of course, they need to give us information, mostly almost in real time. Yeah. So, and at the end, if we're talking about the drug substance process, we need to also, in this case, to have so-called real-time product release. And today I would like to present us our experiences, which techniques we see that they can help us in that matter. So, of course, the PAT is not only inline process monitoring, it's also process understanding. So of course, with that, you can assess that with the modeling techniques, so of your process. But at the end, these things needs to be connected together with the third pillar of a PAT, which is basically control. So how to use the analytical outputs for understanding, to have the instructions, basically, how to adapt and what to adapt for each process steps to ensure predictive control. And today, yeah, I will start with this first pillar, so PAT tools, which we can have for that. So just overview of let's say different process steps and where these tools can come in. Yeah, so we don't need to have on each process step or before or after process step all of the techniques to ensure the tight control of the whole process step. So on the top you can see currently current DSP process, so the discrete batch fed batch processes, but during our Horizon European funded project, we assess different technologies for going to the fully integrated continuous DSP. For sure, there are different approaches, like I already mentioned, and with that we also have different outputs, which techniques can help us, to ensure that. So mostly we come out at the capture step, so the first. For our map processes, the capture step is the first one, is the most important one. So majority of today's talks will be focused on the capture step, and of course we still need to control polishing steps between the whole sequence, which will end at the UFDF step, so the final concentration and the buffer exchange step, where tight control of the target formulation concentrations are really important. So I already mentioned, so what we're looking. So we are basically looking the inline techniques. So if we go from the, let's say, different approaches for the sampling, so how the PAT tools can be incorporated in your process stream, we have different terminology. So currently, and the majority of analytical work is done offline. So of course there are many bottlenecks. They're not continuous. They're not real time. Basically, you need to collect the sample, and yeah, you need to wait to get the analytical results. And this is not the way. That when you're really in hurry to get, let's say, fast responses, to use this kind of analytics. Of course, you have also the at-line, online, and inline. And if we go back to the past, the techniques basically developed in this way. So we know like at-line approaches where we have kind of bypass from your process stream, but after the measurement, this goes directly to the waste, so it doesn't go back to the process stream, in comparison to the online methods where you still have bypass back to your process stream. But in that case, there is the risk that you will contaminate your product or do something with your product, and also this disrupts your process stream, your process. But at the same time, you are never sure that what is measured here it's also completely representative in the real process stream, in the, let's say, tubings from the process on the downstream. But on the other hand, there are the inline approaches where everything is measured directly in the process stream. With that, if we have fast enough techniques, they can be gives us information, the results in real time, or let's say fast enough that we can have some control and response time to do something in the process at the right time. Yeah. At the same time, they can be non-destructive. It doesn't mean, because some of the technologies they are not ready for that, but I'm sure that, which I will show today, that these techniques really will develop, that we can be completely sure and validated that this is non-destructive for the process stream. And of course, with that, that's basically the only representative way how to have the fast measurement frequency. So basically, I briefly explain here the selection criteria which we are looking on. So we are not looking on these kind of techniques because we have these online HPLC systems, because they are not inline. So today I will briefly show you the inline techniques. So just shortly, what are our quality attributes which we are looking on? So of course we have different and many quality attributes on the downstream. So majority, and of course the most important one, is product concentration. So with that, like currently we use like UV-Vis methods or affinity liquid chromatography assays. And then when we go to the different charge variants, glycoform, oxidation, so we have many different techniques. The same is when we would like to monitor our impurity profile, so like aggregates, particle formation, fragments. We have like a tool set of different techniques, which they are not completely non-destructive. They're completely destructive, basically, on your sample. So there is already one limitation from that. Of course, for the UFDF, it's also important the excipient concentration. When we are like really at the beginning, we also have the residual DNA measurements, leach protein measurements. Of course, during the whole downstream, we also need to monitor host cell proteins. But these techniques which are mentioned here, they're really inaccurate. Yeah, basically, the accuracy is quite wide, yeah? and that's also the problem. So for chromatography, I will start briefly from chromatography. So if you look from the point of view of protein A capture step, so the first chromatography step on the downstream, for traditional downstream sequences, the most important is the concentration, which needs to be controlled, especially when we go towards the continuous processes, where we need to, let's say in the multi-column concept, we need to control the loadings between the different columns and so on. But on the other hand, we need to control and monitor mostly like impurity profiles in the elutions. So like aggregates, fragments, charge variants, host cell proteins. We all know that the host cell proteins, that's quite broader range of different proteins which can be HCPs, and it's really difficult to measure today. So, but on the other hand, we have the ultrafiltration, down filtration, where it's more focused on the, of course, again, the concentration, but also the excipient concentrations, like main buffer concentrations, like sugars, chelators, and so on. Yeah, and these concentrations needs to be really in tight control, especially important then for the further drug product processes. But at the same time, we would like to assess the impurity profiles, like the aggregates, fragments, and also the particles. So when we start to when you identify different PAT techniques, you basically end up with like a multivariate tools for which you need kind of statistical analysis to get some output, but you also have more simpler univariate sensors, basically, which gives you usually one attribute at a time. They're simpler, they're already used. And that's on the one hand really easier, but on the other hand, doesn't. You need many of them. Yeah, so the multivariate techniques can also gives you more results at the same time. So if we look, we have like UVV spectroscopy. Most spectroscopy methods are in this region. And we found out that the statistical methods really develop in the way that can be already user friendly and accessible for majority of customers or who they will use them. The techniques are also automated, especially nowadays. The focus also in our company goes in this direction. So. And if we go, so you see, when we start like with the infrared technologies or UVV technologies, and then if you compare to the Raman spectroscopy technologies, you can see all the details of your measurement of interest, how far you can go with them, and mostly this part of techniques is really interested for us. But the question still remains: is the technology already ready for that? So briefly, when we are talking about the PAT chromatography steps. So from the upstream processing, the harvest go directly to the protein capture. So then we have some impurity clearance, and then this impurity clearance is further on polished on the polishing steps. So when we're talking about the protein A capture, so at the time when it's eluted after this protein A the protein A column, so we have this kind of signal output from the existing UV sensors. And of course, we would like to see what's inside, so the specific component of interest. And here is our protein of component of interest. So from the UV signal at 280 nanometers, we cannot do that. And with that, we will need to slice over and see what's happening. So for that, we assess the multivariate measurements. And when we are talking about the after the loading phase, so like what's happening on the elution, so when we released our product, let's say mostly cleaned, this goes again to the polishing steps, and then again the elution is collected. We have this kind of signals, and of course the existing UV sensors are not giving us what we would like to have. So there are different techniques. One of them are already available in the market, so you can use different number of UV sensors. So in this case, let's say you can measure UV signal, let's say at different time points, and with that you can deconvolute from that spectra what's happening. So in this case, from this technique, if you have, let's say, the UV sensor before and after the column, you can calculate, the breakthrough of your protein. But on the other hand, you don't see exactly what's happening inside, so where the components. So this is just for a fast control. So we assessed with the collaboration during the Horizon project what's really happening inside. So we offline collect the measurements from the samples in the fractions, and then we get this kind of output. And again, we use the UV signal and measure the whole spectrum. So at that time, we get this kind of dependence, and with that you can model and get the outputs. So this is from the UV, but we already know that the UV doesn't have, let's say, the full spectrum doesn't give you really details. It's mostly like what you will get from the UV. So let's say I briefly showed you before that the elution peaks are usually at high concentrations, and because they are at high concentrations, you cannot get the whole shape. So we also assess this variable path length UV spectroscopy. So it's so-called UV spectroscopy, in which at different path length the absorption is measured, and with that you can directly assess the concentration even at high concentration. So the concept is the same, just the way how the UV spectra at different times are assessed is different. So on the other hand, I showed you the UV. So the UV has, let's say, one limitation, which is that it doesn't see, let's say, specific components inside. You only see the overall UV signal. So what we really find out is the Raman technology. So with the Raman technology, we needed, let's say, to start to assess this technology working on the flow cells, because we need the flow cells. In the past, we used the probes for bioreactors, because mostly the development started from the USP. But with these new flow cells, we assess the new areas, and of course, again, we had some limitations about the volume needed for that. And how these flow cells are scalable. And when we need to develop the different projects and process steps at like lower scale for the capture step, we don't have so big tubing, so we need smaller flow cells. So we started to investigate with different partners kind of enhancement approach from the back reflector. And with that, we basically assess, let's say, the samples at low volume to even increase the Raman output. And of course, with that, this approach allows us that we measure faster. Okay, now we are the Raman outputs and the frequency of measurements started to be comparable to the UV methods, which are fast, and they are comparable now. And the output, of course, gives us the response times needed for us. So here I will just briefly see how we are working, where you can place the flow cell. In this case, the flow cell is after the output, after the chromatography system between, let's say, offline fractionator, and of course, we need to develop the models. And because the Raman technique, we can see then the different components. So that's why we see this technique as the multi-attribute techniques, so that we don't need so many sensors. And now we are on the right flow path, so to see, let's say, which techniques and the lowest amount of the techniques we need to ensure our vision. So then I will stop with the PAT for the ultrafiltration diafiltration. Again, with the assessment of these techniques, which I already started to show you, we find the possibility to go towards the continuous process steps. So in which the concentrations are varying based on the previous process steps, and tight control here is really important. So, for example, slow spectroscopy gives us this kind of inline outputs. So basically, we managed, and we can measure like about 100 grams per liter in a fast possible time, the concentration, and with that we can really assess really at fast time like the control of the process step. So if we compare them, then we also have the Raman, which is basically the fingerprinting method. So with this Raman, so we again repeated the procedure. So you measure the whole spectrum and do the modeling. And with that, again, we can get like a product concentration as the output, but at the same time, we are now assessing how many different aggregate quality attributes we can assess. But at the same time, there is also one technique which came out. So for the particle size distribution measurement, so in our Horizon project, we also assess the FBRM feasibility technique. Also, we were working with flow cells, and with that, yeah, we can get kind of distribution like you see here, yeah, and that's the whole point behind. But then the question still remains, okay, what to do with this kind of measurement? So because anyway, we will filter out after the UFDF the particles, but the techniques exists. It's ready to be inline, and let's say it's still on our roadmap for further alignment, let's say, with the drug product. So for here, I will conclude for today. So I said for the control of process and product attributes, at least we see as our vision that we need tight control supported by the inline PAT techniques. Of course, this is the first requirements for the control of continuous processes. Not only requirements, it's solid foundation, and at the same time these tools can improve our process knowledge during the development. So basically to have the eyes to see what's happening and understand and to build up our mechanistical process modeling knowledge. So at the end, I will acknowledge our collaborators and our colleagues from different partners and also from our company. And at the same time, yeah, I will thank you for your time, and please feel free for any questions later on. Thank you.
Thank you very much for that, Dayan. Now, before I introduce our next presenter, Marianne, once again, I would like to remind our viewers that there will be a live question and answer session at the end of both presentations near the end of this webinar. So please feel free to send your questions at any time during the webinar via the questions tab, and that's located directly below your webinar screen. I've also seen a question or two about whether or not this webinar will be available once it is finished, and I can say yes, it will be available on demand. There'll be no need to download it because you can just follow the link again and it will take you back to view this webinar on demand. Now, please allow me to introduce our next presenter. Marianne Quayler is the Life Science Product Manager at Kaiser Optical Systems, Inc. She has been providing application support for pharmaceutical implementation of Raman spectroscopy for 15 years at Kaiser, with recent years devoted to advancing bioprocess applications of Raman. Marianne holds a Master's of Science degree in Analytical Chemistry from the research focus in chemometrics and process analytical technology. She also serves as a subject matter expert for the USDA. Sorry, let me just backtrack a little bit. She holds a Master of Science degree in Analytical Chemistry with research focus in chemometrics and process analytical technology. I'll now be handing over to our next presenter. Welcome, Marianne.
Thank you, Marcus. And thank you, Dahan, for the great overview of your research into PAT for downstream. I'll be sharing a summary of Kaiser Raman success stories and bioproducts and how industry's experience with Raman PAT in upstream are influencing the outlook for Raman PAT in downstream. Raman has clear advantages over other spectroscopies or alternative PAT tools for bioprocessing. It's well suited for a very wide variety of inline bioprocess measurements due to its insensitivity to water and its specificity to both chemical and biochemical properties. So it's a key enabler of real-time bioprocess monitoring control, which drives advances in optimization and automation. It's been 10 years or so since the first publication was released on the use of Raman to monitor an industrial cell culture process. Following that, there was an emergence of studies that tested Raman's scalability, robustness, what all it could measure from a bioprocess, etc. But in recent years, Raman is no longer viewed as a potential technology for biopharma. It's one that is driving real change. So there's been an explosion of literature that showcased biopharma's adoption of in-process Raman in recent years, especially for optimization and analytics in development and process control in manufacturing. There are a ton of industry success stories, and most of them are readily available. If you are new to Kaiser Ramen, I encourage you to check out these previously recorded online seminars, which are available from our website. It's a great way to get caught up really quickly on the many advancements that are happening in this industry with Ramen. So why is Raman spectroscopy so popular in biopharma? Well, Raman probes can be inserted directly into bioreactors, fermenters, or flow paths to measure multiple process and quality parameters with a single inline probe. This is a list of the average Raman measured parameters found in publications, but the list is not all-inclusive, and not all of these components can always be measured. Detection and quantification levels are subjective to the specific process or type of samples. Other things that can affect Raman's quantification capabilities are how quickly you need a result, if your Raman system and probe is suitable for your application, and the accuracy of the reference samples used to train Raman models. But if we narrow down to one proven application and trusted Raman equipment that's suited for that application, these unknowns become much easier to address. Kaiser has many years of experience in upstream, so we can share best practices for typical bioprocess use cases of Raman. And keep in mind that Raman sampling and Raman methods should be adapted to fit the problem you are trying to solve. One-size-fits-all Raman is not a good solution for complex applications, especially in the cases of high-value measurements. I wanted to stress this in relation to Raman for downstream. Today's probes may not be very well suited to downstream's very fast response needs, but that doesn't mean that Raman's not an excellent solution to solving many downstream measurement problems. Kaiser excels at tackling the most challenging PAT applications and advancing our optical technology to achieve Raman data that is optimized for the process and is repeatable and reliable. Raman's successes in upstream have been one of the main drivers for PAT in biopharma and a key enabler of advanced bioprocess control. The capability to continuously measure, adapt, and control these extremely complex bioprocesses leads to decreased variability, decreased waste and development time, and replacing offline and delayed analysis with real-time in-process analytics drastically reduces sampling bottlenecks and enables immediate response to process deviations. Bioprocess operations become much more optimized and more efficient and are able to produce higher quality products. And all of this ultimately leads to better, faster therapeutics and vaccines to improve patient outcomes. The majority of Raman applications in biopharma to date are focused at the bioreactor. Kaiser offers a portfolio of analyzers, probes, software, and services that are optimized for bioreactors in lab and GMP. Since our launch into biopharma around 10 years ago, we've continued to improve and expand our portfolio specific to the needs of bioproduction users. Kaiser Ramen is known for its trusted performance and scalability, and we're committed to innovation as well as advancing our bio portfolio to meet the industry's rapidly evolving needs for PAT. Let's look at Raman probe technology. This is one of Kaiser's specialties. Our probe options have grown to include a range of bioprobes optimized for various different bioreactors. In addition to bioreactor compatibility, another major technology advantage for Kaiser is our unique window material. Most other Raman probes use sapphire. In fact, this is the material that Kaiser uses in every other industry but biopharma. But we found early on that sapphire peaks drastically reduced the usable range of peaks in a bioprocess spectrum. So we decided, we looked at different materials and selected one that was much better suited to biologics. Kaiser windows have a low, predictable Raman background and no interfering peaks. Our window peaks are very, very small compared to sapphire and do not mask peaks from bioprocess components of interest in upstream or downstream. Also, our probe materials and collection zones are the same across all of our bioprocess probes, whether that's reusable, single-use, micro, up to large scale. Plus, they include self-alignment and simple calibration protocols for unique model transfer capabilities. Other Raman instruments and probes have a more difficult time with method transfer, even across system to system and probe to probe of the same scale and type. And I want to point out that the data transfer across the Kaiser probes shown here are not in a particular order. Our probes were designed for data to be completely interchangeable from each probe type, assuming best practices are used for data collection and modeling, that is. We understand that it's extremely important that predictive Raman models are not limited to one scale or type of bioreactor. Models need to easily transfer to different analyzers and probes, as well as different bioreactors, whether that's across the hallway or across the world. So that summarizes probes. Let's switch now to looking at other components of a Raman system. If we look at industry trends, it's clear that biopharma is undergoing a transition toward investments in data and automation. This is not unlike the wave of digital transformation that's reshaping just about every other manufacturing industry. To support this trend, Kaiser has undertaken major updates to our analyzer and software portfolio. We've transitioned to embedded analyzers where the hardware and software are unified for a fixed purpose of collecting and communicating Raman data. This aligns with Industry 4.0 strategies such as built-in intelligence and improvements to big data handling. With our embedded Raman RxN systems, external computers have been replaced with a dedicated controller located inside the analyzer box. And we've made many other design and operational changes and improvements, such as smarter diagnostics and more robust, safer fiber optic connectors. We've also optimized our analyzer functions and communication protocols to simplify sharing of data and controls. This allows us to majorly simplify integration to external devices and other software platforms. Our Raman RxN portfolio now consists of only two main analyzer types, the Raman RxN 2 and Raman RxN 4, which both come in 1 or 4 channel options. The 2 is most commonly used in lab scale. It can be cart mounted and wheeled around the lab, or housed on a benchtop or near a benchtop. For manufacturing scale, the Raman arcs in 4 can be stacked in server racks for remote location, or housed in a NEMA 4 enclosure for location close to the process. So the inner parts and performance across these analyzers are identical. Just the exteriors and accessories are optimized for the intended environment. Raman Runtime is the control platform and user interface for our embedded systems. Whether you're at the analyzer, controlling via touchscreen, or accessing runtime through a remote connection, runtime is used to calibrate the system, configure your Raman collection, apply predictive models, and transfer data and diagnostics to external devices, such as a DCS or a SCADA system. Raman Runtime is very intuitive for novice through advanced users, but also all of our analyzer and probe controls can be triggered via standard communication protocols. In the case that your site already uses a preferred PAT platform like Cqas, SciPat, or Process Pulse. In this case, OPC UA offers the most comprehensive bidirectional option, but basic functionality can also be carried out via Modbus. And we also work with many different integrators to create OPC UA pre-configured adapters to our systems. These pre-configured and pre-tested communication saves the end user a lot of time and resources, especially when it comes to validation and GMP. Software connectivity is a critical component when Raman is integrated into other hardware products, especially. An example of this is with our recent collaboration with Sartorius for the BioPat Spectra platform. Here, Kaiser Raman controls are fully integrated into the Amber software. Amber tells the spectrometer when a sample is in place, a Raman spectrum is collected, and then data is transferred to Amber software and organized along with additional bioreactor data. This is repeated to collect Raman spectra from all bioreactors running in an Amber 15 or Amber 250. So smooth device-to-device communication is imperative to success of automated platforms like this one. Kaiser and our customers are really excited about this collaboration with Sartorius, but I'm not going to spend much time on this product today because I've already covered it in a few other online seminars earlier in the year. But the highlights are, 1, integration with Amber enables easier, faster, more price efficient, and more robust Raman model building. High throughput development with Kaiser Raman supports early stage QBD. And since BioPat Spectro is also compatible with Biostat STR, scale up to single-use manufacturing is much more efficient. The amount of high-quality Raman data that this product can generate very rapidly, I think is going to make major contributions to in-process data analytics across the biopharma industry. The addition of Amber and Biostat STR compatible probes really rounds out our upstream bioproduction portfolio. So I wanted to highlight this and the many advancements made by Kaiser, our customers, as well as our partners in upstream bioprocessing, so you can understand where we're starting from for transitioning Raman into downstream. Through our upstream experience, we've built up a ton of subject matter expertise for developing, producing, and supporting Raman-based PAT solutions in biopharma. The Kaiser Raman team has extensive experience with in-process Raman spectroscopy across a wide variety of industries and applications, but recent years have been heavily devoted to the biopharma industry. Our awesome people and their dedication to our technology and dedication to our customers are one of the many things that makes Kaiser Raman stand out. These are some of my colleagues having a little socially distanced fun conducting our first fully virtual factory acceptance test. This happened a few weeks ago and was really well received by the customer. We expect this virtual FAT offering to be a popular service even when travel resumes, because it saves so much travel time and cost. And this was a nice, simple example of how Kaiser wants to help you reduce the complexity of implementing PAT process equipment and methods. Our experience, training, application, and qualification services let our users focus on their core business and quickly realize high value and rapid return on investment from their investments in Ramen. So where are we with downstream? I really enjoyed Daehan's take on some of the challenges, but also the many opportunities for adapting Ramen to downstream, especially for connecting continuous bioprocessing steps. We have several customers who are already implementing Ramen monitoring and control in several DSP operations. Some of these studies are available, such as the Pfizer one referenced here, but many are yet to be published. However, there is a lot of Kaiser Ramen activity happening in downstream. We are actively collaborating with industry experts to ensure the downstream products we're developing meet or exceed the market requirements. So while our end goal is to optimize Kaiser Ramen technology specifically for downstream needs, in the meantime, some companies are already realizing Ramen-based control in downstream using our standard bioprocess probes combined with an Endress Hauser flow assembly. The flow cell shown here is commonly used with Endress sensors for chromatography and filtration operations. Our 120 millimeter length bio-optic fits into this flow cell assembly without modification, and the sizes range from 1/4 inch up to 2 inch tri-clamp connectors. This setup does have some limitations with speed and detection limits for early DSP steps, but it's a great option for fast inline measurements of protein concentration in later downstream operations like UFDF. It's also just a nice option for getting started on investigating inline Ramen opportunities for flow paths. If we look collectively across upstream and downstream and how Ramen technology is contributing to biopharma, the overall trend is that it's driving PAT and digitalization initiatives due to the uniqueness of where and what it can measure in real time. Ramen spectra collected continuously from a bioprocess are often transformed into simple univariate outputs like a glucose concentration in upstream or MAB concentration in downstream. These univariate outputs are easy for existing data systems to read and to act on, but one Ramen spectrum actually generates thousands of data points that provide a wealth of in-process information. It gives you a fingerprint of your process, but there's data infrastructures aren't quite able to handle this massive amount of data quite yet, or the multitude of analyses that can be used to extract the most important information from spectrum. But we're seeing a lot of improvements to data analytics, which are already drastically helping to fill these gaps. So the future for Ramen PAT in upstream and downstream is very promising. Knowledge about new bioprocesses can be acquired faster through real-time measurements. These processes can become leaner and more tightly controlled. And when they are transferred into clinical and commercial manufacturing, they'll be highly automated, productive, and produce higher quality biologics and biosimilars. This is already happening now, but on a smaller scope than it could be if there was widespread improvements to the digital landscape in biopharma. With continued advances in Ramen technology and to the data it produces, Ramen probes could become as common as a pH sensor, but deliver an extraordinary amount of bioprocess intelligence. Some of the major areas Raman is influencing are summarized here. The benefits to product and process development are probably the most obvious. Increased process understanding leads to optimization and the development of more efficient, data-driven processes. Then in production, industry can move away from following recipes to utilizing smart automated controls. It's already well proven that real-time glucose control by Raman in commercial upstream manufacturing drastically increases productivity. Extending that to many other CPPs and Cqas measured inline in GMP has huge potential to shift how biopharma does manufacturing. And Raman's benefits to tech transfer are often overlooked or underutilized. Raman spectra from raw materials and preparation of media and buffers, as well as spectra from throughout the course of an entire process, provides a compositional fingerprint at each step. Comparing Raman spectra from starting materials at a new scale or location to an expected Raman response provides a simple quality check when you're scaling up or scaling out. And all of the above are especially important for cell and gene therapies to get production right the first time. So I've covered a lot in a short time, but hopefully you've grasped the common theme that Kaiser, together with Endress Hauser, are committed to helping you advance your bioprocess measurements. Kaiser Raman technology provides inline, real-time monitoring and control of many different properties and is applicable to many different bioprocessing steps. And Kaiser stands apart from other Raman options through major technology innovations and our industry know-how. Kaiser Raman is proven as a PAT enabler from development to manufacturing and traditional to single-use platforms, as well as batch to continuous bioprocesses. And now we're seeing that advances in upstream are heavily influencing in-process analytical measurements in downstream to ultimately make quality biopharma products more consistently and more efficiently. With that, I'd like to again thank Dehan and also my colleagues at Kaiser and Endress Hauser. Thank you to our listeners as well for your time and attention. Hopefully you learned something new about Kaiser Raman today. All right, I'll pass it back to you now, Marcus.
Thanks a lot for that, Marianne, and welcome back to the presenter screen. Yep. Okay, so before we jump into our Q&A session, just one last quick announcement to our audience members. You can still send your questions if you want to via the questions tab directly below your webinar screen, but please state in your questions which presenters they are for. That helps us a lot. So we'll jump straight in, and our first question today will go to Dehan. So have you looked at microfluid modulation spectroscopy, MMS, for Redshift Bio? How effective is the technique as compared to Raman technique?
No, not yet. But yeah, for sure. Yeah, I will be interested, to look on this technique also, to see and compare.
Yeah. Before we move on to our next question, is there anything Marianne would like to add to that answer?
Not for that one.
Okay, great. So our next question goes to Dehan once again. I have seen that PO2 sensors. Where do they use it?
Yeah, this is basically it was a general slide, so different techniques were, let's say, on this slide, and of course it was not differentiating if it's for DSP or USP, so this is not usually used for the DSP, so we don't use these sensors on the DSP, but for sure on the upstream, this is then the different story.
Thanks a lot for that. Any additional comments from Marianne before we move on?
No.
Okay. Next question goes to Dehan once again. What Cqas could be effectively monitored by inline Raman spectroscopy?
Yeah, the first one is the concentration, the most important one, concentration of our protein, product of interest. then excipient concentrations for the UFDFs, like already mentioned. So these kind of really different components, can be really effectively measured by the Raman, especially if they are not at too low concentrations. But when we're talking about the aggregate, so we are assessing now these things. Yeah.
Thanks a lot for that. Any additional comments for Marianne before we move on?
Nope, that's the primary ones. Again, still actively researching what can be measured by Raman and downstream, especially when we start talking about signal enhancements compared to what we're doing in upstream.
Great, thanks a lot for that. Our next question, I think, goes to Dehan once again. However, please feel free to nominate Marianne if you think it's more relevant. Are you collecting GMP data or not GMP data during process development?
So during process, no, not GMP data. So mostly for the development, lab scale, pilot scale. Most of the time when we start to develop the techniques or flow cells, in the case of the downstream, we're not ready for such a large scale. But some techniques, they were already assessed in our manufacturing sites. But of course, we started at the development first.
Great. Any additional comments, Marianne?
No, that's pretty typical. Most customers start out non-GMP, build up models and application know-how and development. And then when it comes time to transfer to GMP, sometimes users will take the extra step of collecting additional data on GMP validated systems to use for their multivariate models, but that usually depends on how they're intend to use the models. If it's just for monitoring, the validation of models is less important. If we're talking about control or especially replacement of quality measurements, then the more rigorous steps need to go into the way the data was collected and how the modeling was created.
Great, thanks a lot. Our next question goes to Dehan once again. Do you have issues with fluorescence interface when you use the Raman for monitoring the protein A capture? And how do you overcome these challenges?
So on the samples after the protein A, basically we never monitored the fluorescence, especially not on the UFDF. But we are monitoring that, and from our experience, we really never had problems with that. But there are techniques, if in the case, especially from our upstream colleagues, we can learn on that. But we never had this kind of problems for our samples. That's true.
Great. Any additional comments, Marianne?
Yeah, downstream samples are much cleaner than upstream when it comes to the Raman spectra. So no, we haven't seen any fluorescence problems in downstream quite yet. And that's, we're always using 785 nanometers typically for downstream tests in biopharma. We do have another wavelength option that further reduces fluorescence interference should it occur.
Oh, shit. Sorry.
Muted. I think I may have that's okay. I may have gotten muted. Not sure when. But I was mentioning, that if fluorescence were to occur, we do have a longer wavelength option that's closer to 1,000 nanometers that has completely rejected fluorescence in upstream bioprocessing where it's been a problem. Oh, now you're muted, Marcus. Let's see. I can chat with him.
Better? Can you hear me now?
Yeah, there you go.
Okay, sorry about that. So our next question goes, I think, to you this time, Marianne.
Okay.
What kind of process parameters are required in a downstream process?
As far as what can be measured by Raman, I think that was answered previously. Protein concentration is the obvious one, buffer excipients, and then we're looking into quality markers like aggregation, host cell protein, etc. No clear rules and limits to that yet. Again, a lot of that will improve as we're continuing to enhance Raman's capabilities in downstream to collect to lower levels and faster.
Great, thanks for that. Any additional comments from Dehan before we move on?
Yeah, basically, Marianne really addressed this question pretty well. It's not clear. Yeah, some of them are, let's say, really needed for the control, like the concentration of protein. But at the moment, they are more meant to be for the monitoring purposes. And when we will have this data and predictions, then we can establish the strategy how to use that, and which are basically really requirement based on our application, so what you would like to achieve.
Great, thanks a lot for that. Our next question goes to Marianne once again.
Okay.
If there are peaks interfering with the API peaks at spectral information for Raman spectroscopy, these will probably affect the PAT models. How can we overcome this interface if there could be potential solutions?
Well, typically you're not using univariate modeling in these applications. I don't know of any univariate models for bioprocess spectra. It's so complex. There is a lot of overlap. So you're typically using chemometric methods, multivariate tools like PLS, etc., to help you extract the most relevant information from your different components of interest. So overlap is typical, and it can be handled with chemometric methods.
Great, thanks a lot. Any additional comments from Dehan before we move on?
Yeah, that's a good point. Yeah, we use the multivariate method. So, and of course, it's really depends on the Raman settings. The one side, how long you will measure. So we are now assessing that with our progress with the flow cell development and enhancement of the signals. To even increase the sensitivities to see how far we can go and where are really the limitations from the technique. So this is one approach. The second one is kind of hybrid modeling approaches, which we are also exploring, to using the process models connected and coupled with the Raman and to assess the prediction about that. Yeah. So there are different approaches, completely like a process data driven, or let's say from the technique, like I already mentioned, to see how sensitive and how fast and how long you can collect the data.
Great, thanks a lot for that. Our next question goes to Marianne once again. Are these applications generally focused on trending different species, or are multivariate models applied to the data to generate concentrations for quantitative analysis?
A little bit of both. I'd say the majority of customers jump right into quantification. They want to measure key process parameters, glucose, lactate, etc., and upstream, protein concentration downstream, and they want to do that in a quantitative way, not just trending. Although trending your process has excellent potential, again, for tech transfer, looking at process deviations. In that case, you're really looking at the process fingerprint across the full spectrum and all the things changing at once throughout your process. So trending is used and probably should be used more, but I'd say most of the time quantification is happening to basically transform the spectra from a multivariate output into a single output, such as concentration values.
Great, thanks a lot. Any additional comments from Dehan before we move on?
Yeah, I will just confirm. Yeah, so the quantitative outputs are really our first interest, because this is easy to assess, and we are working on that, and to see which kind of attributes can be measured quantitatively, and then to see then the different approach, like Marianne already said, so the trending, relative changes, and these kind of things. That goes to the previous answer from the previous question, for these kind of more complex attributes which are hidden inside of our measured spectra.
Great, thanks a lot. Our next question goes to Marianne. What would be an ideal setup if the sample solution is going to be frozen during experiment? Should the Raman probe be inserted into solution, or placed above solution, or placed on glass and collect through glass during freezing and data collection?
I'll have to defer that one. I don't have a lot of experience with frozen samples. I know we've assessed them before, but I couldn't advise on the best placement on that, so I'll have to take that one offline.
No problem. And for questions that come in later or are not able to be answered today, don't worry. We can follow up afterwards with these questions by email to the audience members who submitted them. So our next question, unless, sorry, before we move on, Dejan, do you have any additional comments to this question before we move on?
No, because I also don't have any experience, to measure directly. I mean, I suppose with kind of non-contact optics ways, to measure, with the touching. But of course, this really depends on the material of your container and so on. So, how to assess these things. And of course, you also have the interference for this material, so you need to be careful. Yeah.
Great, thanks a lot. Our next question, I think, goes to the both of you. So there's a comment. Hi, Dejan and Marianne. Thank you for the great presentations. The progress in the area is impressive. Now for your question. As an offline analytical scientist, I'd like to understand how our regular analytical controls are transferred to inline assay format. So number 1, instrument collaboration. 2, SST and QC analysis, like sample bracketing and with controls. Number 3, QA or audit data reviewing. Marianne, perhaps you'd like to start?
Sure. It's usually a staged approach. Immediately replacing offline methods with inline ones happens rarely at once. Starts out with monitoring, making sure the measurements you're getting inline are matching your offline approaches. And then depending what your end goal is, whether it's just inline monitoring your process or control of your process, or actually completely replacing the offline analytics with inline ones, there'd be, I guess, various validation steps you would need to take in GMP to reach that state, especially for real-time release. This is being done regularly in small molecule biopharma. I don't know of any instances of real-time release in biopharma using spectroscopic analysis instead of offline analytics. But hopefully the industry will get there someday. But I'd say in general, it's a staged approach. Start simple with some of the primary components of interest and prove that your inline measurements are matching your offline, and then move on from there to reduce your offline sampling and eventually eliminate.
Great. Any additional comments from Dejan?
Yeah, basically, we are also assessing, how this approach will be, and I think PAT as a concept and how to approach this. I think the FDA already started to, establish this from the small molecules. So of course, you will need for validation of your prediction models to use kind of complementary offline methods. So for which you have already validated assays and the results, and to compare it at the strategical times and how. So these kind of things, of course, as I mentioned, it was already mentioned, is really stage approach. Yeah. So even in the now where we are approaching and evaluating the technology during the development, we follow all of the principles. So we start with the calibration. We have the reference standards to see if something is wrong with your instrument at first. So the procedure it's going in hand to hand with the offline techniques, but of course, because it's the inline and for long-term measurements, you need to assess this a bit differently. But for sure, these things are already starting to address from a small molecule world, and of course they will be transferred to the large molecule worlds. Yeah.
Great, thanks a lot for that. Our next question for Marianne. We now generated a glucose model from several runs using SIMCA, SIMCA. The question is, how can we integrate this model in the Kaiser system RxN to lab scale for the next run to obtain live glucose values?
Sure. So we have several predictors embedded into our Raman runtime platform, and SIMCA is one of those. So kind of depends on what software the user is using, whether it's a legacy software or our new runtime platform. But with our runtime platform, it would be as simple as obtaining the SIMCA real-time license if you don't already have that, and then uploading the model to your runtime system.
Great, thanks a lot. Any additional comments from Dejan before we move on?
Yeah, basically I can confirm. So how we do it, like Marian mentioned, yeah, we use Cqas if we use the platform from Kaiser. So runtime in our case, but yeah, there are also other possibilities like to automate the export from the instrument somewhere else, and then you have then the different options. But this really depends on your strategy and platform and this data management.
Thanks a lot for that. Marianne's next question is, how did Kaiser prevent probe fouling?
We don't have much probe fouling in the bioprocessing world, thank goodness. But we are still looking into ways to always decrease that in case it does become an issue. One of them is optimizing, again, the probe for the intended use. So we have a lot of experience in upstream, and we've been able to design our probes in a way that we don't see much probe fouling issues. And most of our opportunities in downstream will likely be non-contact, which eliminates the probe fouling issue in downstream.
Great, thanks a lot. Any additional comments from Dehan before we move on?
Yeah, we never experienced, and it was that we are now in the way to have non-contact approach. So all the flow cells, basically we get rid of the probes. So either we use the non-contact optics or directly connection to the fiber head. So the progress in this area goes really in this direction, especially for the downstream, to get rid of all of these kind of potential problems.
Great, thanks a lot. Our next question then goes to Dehan this time. Have you already had a chance to test mid-infrared spectroscopy in DSP? And if yes, what are your findings?
Not mid. I tested the near one. So at the time being, this was quite years ago, the technology was not really ready for that. So we tested it offline. So of course, the results, were like using this infrared technology. Of course, at the time being, we let's say leave on this technique and goes to the mostly the techniques which I already mentioned. But yeah, I saw different progress in the development of these infrared techniques, and for sure our vision is also to assess different techniques like ER. We'll mostly see it as a complementary method, because as already mentioned, of course we have like Raman and the UVVs, but they are all cons on different sides, so they're basically complementary. And what we would really like to let's say explore is this complementary approach, to using some of them or many of them to get really bigger picture. Yeah.
Great, thanks a lot. Any additional comments from Marianne before we move on?
No, Dehan covered what I was going to say, in that, spectroscopies are complementary. They each have their own specialties, and combining the efforts of some of them, in some cases, will give you better results than just one. And in other instances, you may have advantages for one over the other. So it really depends on the application you're looking at and the problem you're trying to solve.
Great, thanks a lot. Our next question goes to Dehan once again. Do you see mechanistic process modeling working with inline spectroscopy techniques for process control? Is it something industry is interested in?
Yes, of course. We really see it, especially personally. I see the way to go, so the next step, basically. As I mentioned, so we started with the technique alone, so to see how many attributes we can get really reliably with good predictions. But this is also then the goal to feed in and work together with the process mechanistical modeling, which is at the end our goal, because then we can use this for process control. Yeah.
Great, thanks a lot. Any additional comments from Marianne?
I don't think I have anything to add to that one other than that. Yeah, it seems very promising to expand the capabilities of spectroscopic techniques.
Great, thanks a lot. Our next question goes to Marianne. Are Kaiser systems compatible with GAMP5 integrated automation requirements?
Yeah, our runtime platform is developed according to GAMP. Runtime itself is meant to continuously collect and distribute data. It's not meant for long-term data storage. So it does not meet all requirements for 21 CFR Part 11 compliance in GMP. But we work with standard platforms that are used to handling spectroscopic data in a GMP environment. So it's compatible with all the standard platforms being used in industry now.
Great, thanks a lot. Any additional comments from Dejan before we move on?
Yeah, basically that was our, why we went to the runtime. Yeah, so when we started, there was still the IC Raman, but then this new platform gives us this possibility, and of course our main goal is connectability to the whole ecosystem of this external system.
Can you still hear me, Dejan? Sorry, are you okay to just repeat that answer? There was a bit of technical issues getting involved there.
Yeah, sorry. Yeah, basically, I confirmed that because of that, we went to the runtime platform. Some years ago, I think already 1 year ago, because this gives us possibility to connect to this control platform ecosystem. Because at the end we would like to control, but also for the data modeling.
Okay, so sorry, so your answer was a bit broken up there, but hopefully we can follow up with that later by email as well.
Okay.
So for our next question, it goes to Marianne. Is it possible to detect and preferably identify leachable impurities with Raman?
Hopefully leachable impurities would be extremely low, so probably not with Raman.
Okay. Our next question. Yeah, it doesn't state to its full, so I'll ask Dejan first. In bioethanol production from biomass by fermentation process, if Kaiser Ramen used is their huge ethanol recovery rate increased? Can you discuss any case study? So perhaps Marianne would like to take this question then.
Yeah, I mean, we've spent most of our time in biopharma, at least I have. But we have had success in other bioproduction offerings with different fuels, foods, etc. I'd have to follow up with that on the specifics offline. I don't know if there's any case studies I know off the top of my head that can really help answer that question now.
Mm-hmm. Okay, great. Any additional comments from Dejan before we move on?
No, basically no, because I don't have any experience with that. Yeah.
Yeah, okay then. So our final question for today then will go to Dejan. Are there any complementary inline techniques or approaches to enhance current PAT tools outputs?
Yeah, so one approach is, like I already mentioned, if we stick with the same inline techniques, so to go in the direction of process modeling, so-called advanced process modeling, so where these techniques can help and fit this process modeling to address process variability issues. So this is one, but at the same time, yeah, we are exploring different analytical techniques, like already mentioned, so like infrared spectroscopy together in hand-to-hand with the Raman or UV-Vis. So of course we can be complementary in the sense of the different techniques, but we can be also complementary in the sense of modeling, basically to get the predictions.
Great, thanks a lot for that. So thank you very much, audience members, for your questions. Before we wrap up today, I'd just like to ask our presenters if they have any closing remarks. We can go in order of presentations. So Dejan, perhaps you'd like to go first.
yeah, but basically, I really like to thank for your time and that I think you, and I hope that my talk was interested for you, and yeah, I wish success for all of us on this way, if you're working in this area.
Great, and over to you, Marianne.
Yeah, I second that. Thanks for your time, and especially sticking around for questions. There were some great questions. Very happy to be here and to share what we've been doing, and it's great to see some success stories shared by Dehan, and look forward to doing this again. Any questions I wasn't able to answer, or if there's more, just feel free to follow up with me after the seminar.
Yeah, and I would just like to second that as well. So if you do have any more questions, or perhaps we couldn't get around to your question today, don't worry. Please feel free to send your questions over to me or to Marianne or to Dehan. My email address is being displayed below your webinar screen right now, and I'll pass those questions over to our presenters and then get those back to you. So I'd like to take this opportunity to thank our presenters. Once again, that was Dehan Arzenzek, Principal Scientist at Novartis, and Marianne Quayler, Life Science Product Manager at Kaiser Optical Systems, Inc. Thank you, both of you, for sharing your knowledge and your expertise with us. And I'd like to remind our audience that you can view this webinar and other webinars on demand by visiting our website, which is biopharma-asia.com/on-demand-webinars. And if you're watching this on demand, then please feel free to send your questions over to me. And my email address is marcus.orsay@biopharma-asia.com. And we'll get those answers over to you. So thank you, everyone. Thank you, Dehan, and thank you, Marianne. And I hope you all have a great day. Take care now.
Thanks, Marcus. Bye, everyone.
Bye. Bye.